So what happens in a drug discovery process? Let us call it, okay, drug discovery pipeline. Okay, so this is the pipeline. So as you have already had a discussion previously, that there could be two approaches. Let us call it bottom up or bottom down. By and large, what exactly happens is either you are having a already designed small molecule, okay, so that you need not to search for the database. In our case, in this work that we did, I want to again put a this thing. Yes. So in this paper, what we did the wet lab study first. What we did was we started with this particular mononuclear copper, which is having a kind of transition element kind of behavior, and we also found out that this particular architecture of the molecule will have some biological property. So what will be the biological property? The biological property is catechol oxidase activity. Okay. So why this oxidase activity is there? Because copper is having a variable valency. If it is if it is already in the oxidized state, the valency will be 2, that is cupric, or it can even be a reduced form, that is cuprous. So this is one thing. So since it is having an oxidase activity, we can eventually use this particular combination of architecture for multiple different reasons. As I have already given you examples, like we can even put it in diagnostic kits, okay. Then we can even put it to act like a redox drug, okay, a redox drug. So can any one of you name a redox drug which is either linked to free radical scavenging or free radical removal? Can any one of you give some examples of such kind of drugs which can act like a free radical scavenging thing? Anyone? Can anyone give us some examples of drugs which are basically used for redox reactions? Let us say glutathione. How many of you have heard about glutathione? Glutathione is a wonder drug which is acting as an antioxidant mainly for liver damage. Okay, so those people who are having a highly intoxicating nature, they will end up with so much of free radicals in their system, which is basically neutralized in the liver parenchyma. Okay, and therefore there is huge stress on the liver parenchyma, and that oxidative stress is reduced by redox drugs like glutathione. So in this study we have identified this activity of this molecule, and eventually if you want to have this catechol oxidase activity in the body, or that is in the biological environment, you should have sufficient HSA binding. Okay, so what happens, as we have already discussed, that your drug like molecule, if something is drug like, it should have sufficient bioavailability. Okay, some of you would already have some idea about it. Then subsequently you will go for a study called pharmacokinetics and pharmacodynamics. Pharmacokinetics and dynamics. Now these kind of studies will give us idea that how the metabolism of the drug will be there in the body and how it will be excreted. Okay, these two things will come to know after these studies. Okay, the metabolism of the drug as well as its excretion. Okay, why excretion is important? Because it will give you an idea what will be the dosage form. Okay, so if it excretes rapidly, then dosage should be very, very substantial. Okay, so it will give you an idea of the dosage form. Dosage form means in which architecture you will use the dose. For example, will it be injectable, will it be like oral? Okay, now what is dosage form? It will give you an idea whether the molecule will be injectable, or oral, or things like that. Okay, so these are different aspects of it. So what we did, we identified this redox type of molecule, and we tried to see how it binds, how well it binds. Okay, so what we studied the binding of the sheaf base with albumin. Okay, so what we did, we have already spoken already. We have studied the binding with human serum albumin as well as with bovine serum albumin. Why? Because if we are having very good binding interaction, it means that there could be sufficient this thing, what do you call, bioavailability. Okay, in human, okay, to act as human drug, or you can also, if the binding of the same molecule is also good with HSA, then you can use it as a, what do you call, veterinary drug. Okay, so that was the idea. If HSA binding is also, BSA binding is also good. Okay, now this, what happens is, I am audible, right? So this is a bottom down approach where you are only playing in silico. So as you can see here, small molecule database. So this is an example of a database. We will directly go to a database which is this thing, a drug like database, or a database which is essentially a complete database of this thing, what do you call, drugs. Okay, or small molecules. You can even consider it as a small molecule database. So I am sharing with you the link of the tool which will help you to use this small molecule database. So this is the first step, as you can see. So either you can use this small molecule database, or you can have 3D structural database as well, or molecular fragment databases are also there. Okay, so what exactly is your work is to filter from a large database what exactly, which kind of exact drug you want to design. Okay, it can happen that the small molecule database itself is not having, okay, just hold on. Okay, okay. So it can happen that your database may not have the exact drug, but it will give you a certain idea. Okay, so and one more aspect of this is you have to also see that whether the small molecule that you are about to design is having some interactions as well. Okay, so interaction studies are also very important. I will get back to that, which is generally termed as this. Okay, ADME and toxicity estimations. So consider that you have identified a ligand or designed a ligand which is very efficient to bind to a particular protein or a particular pathway. It can inhibit a particular pathway. Let us give us an example. So consider that there is a pathway called uric acid synthesis. Uric acid synthesis pathway. Okay, it's a very basic pathway and very common also. So what happens in uric acid synthesis pathway? What happens? All your nucleotides, let us say DNTPs, mostly the purines. So your purines will end up the purines will end up as a metabolite which is known as the uric acid. Okay, so uric acid. Okay, so what would happen if so WHO recommended range of serum uric acid is recommended range is less than 6 mg per deciliter. Okay, you remember this. So this is the recommended range of uric acid. So now from where this DNTPs comes? The DNTPs comes from your food. Okay, mostly this thing animal food, but plant foods are also there. So let us call it food stuff. Okay, so your food stuff could be highly this thing with cells and which eventually give rise to a lot of purines which eventually turn it into uric acid. Now if everything is okay, the uric acid will get excreted from the body. But what would happen if the uric acid is building up? Okay, so if the uric acid is building up, then it will lead to a condition called gout. Okay, now this is not a autoimmune gout, it's a, let us say, uric acid gout. Okay, so it will lead to a lot of discomfort. So what is the medicine? The medicine is an inhibitor of the uric acid, or the structural analog of what do you call purines. What is the name? Who can tell me? Any name? Allopurinol is the structural analog of purine which lead to the closing of this pathway, so that no more uric acid is being formed. Okay, so allopurinol is an competitive inhibitor. So this also you already know, it is a competitive inhibitor of purines. Okay, and therefore what it does, it stops the formation of extra uric acid crystals. Okay, eventually the patient is somehow this thing. So how this idea generated? They were playing with when the pathway was identified, uric acid pathway, then what they did, they identified a loophole. What was the loophole? The loophole was there in a enzyme. So they identified the enzyme. Let us consider that this is the enzyme. Okay, now this is the pocket. Okay, now this is a pocket. Now this is a binding pocket. Okay, so let us consider that there are multiple binding pockets. So this is a binding pocket. So there are multiple binding pockets. Now this binding pocket can accommodate this structure. Now this structure is of what? This structure is of purine structure. Okay, so what we did, we identified an structural analog and we are substituting this purine with this. No, we have to change the structure. Okay, so we are substituting this with this, which is having some analog, some structural analogy. Okay, so structural analog of purine. So what it will do, it will come and bind here, isn't it? It will come and bind here, and therefore this binding pocket will be eradicated. So identification of an binding pocket is very, very important. Okay, so here also, as you can see here, structure based docking, structure based in situ design, lead design, and all such, the whole step, this particular step is dedicated to this, to identify a structural analog. Okay, so now what we'll do is, we'll do a little hands on, so that we can do two things. One, we'll see if a already established drug is there, how it will interact, because this particular area is also very important. So will it interfere with some other pathway or not? Okay. If the drug is good enough to become very, very specific or not. So that study is also very important. So in order to study this, consider that in order to study this particular parameter, just hold on. In order to study this particular parameter, we will use this particular tool. Okay. So I am sharing with you all the tool which is very, very useful, and you will also find this particular tool very important also. So first one was the link of a database, and second we are sending you a tool which can help you to identify if there are any what you call crosstalk between the interactions, okay, or whether there are some similarity between the targets. For example, you have used allopurinol as a inhibitor of uric acid pathway. So what would happen if it stops protein synthesis, or if I'm just telling you hypothetically, what would happen if it stops glycolysis or very fundamental things that are going on in the cell. So it will be highly toxic. So in order to identify that, we will use this particular tool, okay. So one by one we will use this. Let us give a demo. So first and foremost, let us talk about some anti-cancer drugs, okay. So a very important anti-cancer drug is aminopterin, okay. Aminopterin is a very important anti-cancer drug, okay. So I'll talk about aminopterin a little more. So here what is there, when you search aminopterin, it will give you the whole detail of the molecule along with structure and with a particular thing called isomeric smiles. Can you see? So what you are doing, you are trying to figure out if there is a drug, is it interacting with some other vital issues or not, so that you can say that your drug is cleared for ADMET analysis. That means the toxicity is bearable to a limit that it can be permitted as a what you call life-saving drug, okay. So isomeric smiles are nothing but IUPAC designs only, where the bonds are not there and only the atoms are being shown, okay. So we will copy this, we will copy what isomeric smiles, and we will go to this what you call database, or not database, this particular tool. Here you can find one, already the aspirin example is already been given. We will not use it, we will use our own smile. So in the smile tab, you will paste the smile and then search. So once you give a search this thing what do you call order, then it will search and find out the molecule, okay. So now this is the aminopterin. Then we have to click on calculations, okay. So now we have started the calculation, and let us see how good the drug can be for human use, okay. So it has identified some what you call very strong binders, okay. So as you can see here, it has very strong binding with cellular tumor antigen P53. Now who can tell me more about P53? P53 protein, who can tell me more? There are many more, but highest, very important here is cellular tumor antigen P53. So what we are discussing? We are discussing the study on aminopterin. So let us go back. Aminopterin is what? Who can tell me? Any insight on aminopterin?
Anti-cancer drug.
Yeah, that is fine. But what is this pathway? Means which in which way it acts as an anti-cancer drug? Anti-cancer drug is ekdam true. So it is a folic acid analog. So last time also we discussed. So it is a folic acid analog, very important, okay. Now what happens, folic acid is very important for maturation of DNA. So folic acid itself is very important for what? So folic acid is very important for maturation of DNA. So it is important for what? maturation of DNA. Maturation of DNA, and therefore it also helps in regulation of the cell cycle. Okay, it also helps in regulation of cell cycle. Cell cycle regulation. Okay. Now what happens? If aminopterin blocks folic acid, what would happen? Cell cycle regulation will be completely disintegrated. Now in cancer what happens? In cancer, a single cell is metastasized into multiple cells and finally forms a mass of cells. Okay, where at random event cell cycle is going on. Okay, so cell cycle is going on at a random pace, and therefore metastasis takes place. Okay, so this is a metastatic cancer. Okay. So what this aminopterin will do? Aminopterin will reduce the chances of cell cycle to make more DNA, and eventually it will kill this metastasis. Okay, because folic acid is not there, maturation of DNA is not there, and therefore what would happen? Metastasis will be stopped. Okay. Now there is one more important thing you should know. Oh yeah, shit. Okay. So one more important thing you should know. So what is the important thing that you should know? You should know that there is a thing called proto-oncogenes. Okay, very important. So proto-oncogenes are those genes which are also helpful for cell cycle regulation. One such proto-oncogene, most celebrated proto-oncogene is P53. Okay, so P53 basically what it does is it stops cancer. P53 is needed to stop cancer. So here you can see. So what aminopterin is doing? Aminopterin is doing some binding with cellular tumor antigen P53. Okay, and the binding potency is how much? Potency is 31.6 nanomole. So what does it mean? It means that to inhibit a single unit of P53 protein, you should have at least 31.6 nanomoles of aminopterin. Okay, so it means that it is not it is binding with its target, which is nothing but a proto-oncogene, very efficiently, and it is not also what you call disturbing any other major proteins. Okay, for example, if it interfere with production of hemoglobin, then what would happen? The person will survive from the metastasis but may die due to anemia. Or what would happen if it inhibits the electron transport chain in the in the mitochondria? The patient will survive cancer, but it will die due to a lack of generation of ATP or some other abnormal neurological conditions. So it indicates that aminopterin can be treated as a very good drug, because it is looking at its target very efficiently, and therefore this is the preliminary screening. Okay, very preliminary screening, which indicates that it is not interfering with what you call very vital proteins in the what you call cellular system. Okay, so similarly, if we search for one more molecule which is similar only, let us say methotrexate. Okay, methotrexate is again an antiviral drug. What happened? No, the patent came, but just hold on. Okay, so what happens? We are trying to figure out the smiles of this
Sir, what does this
particular.
smile means?
Just 1 second. I'll get back to this. What is a smile? I'll just get back to this. Where is the smile? Okay. Yeah, we have found it. Just hold on. I'll get back. So smiles is nothing but a format of chemical entity. Like FASTA is there, no, for DNA and RNA. So let us consider smiles as a format where C, you can see the C, no. So C sometimes looks like a smiley. So consider this. So this is a what happened? Yeah. Consider that this is a C, okay. So if I make it a C, so it will look like a smiley, right? Understood? So since it is a C, sorry. Okay, now is it a C? And now is it
Yes, sir.
a smiley? Understood? Understood? So this is how they are putting
Yes, sir.
this
Yes, sir.
this particular this thing as an interesting form. Canonical smiles are nothing but the format of chemical formula interpretation where mostly you can see the C, and since C looks like a smiley face, we are calling it smiley. So now we'll go back to the same tool to find out how good this particular smiley is as a drug, okay, or it will do some major interactions. What happened? Just hold on. Okay, and then here, yeah. Now what we'll see, we'll try to figure out that how the interaction is, whether it is good, then it is better. They both act on the same pathway, but we are trying to figure out which is less toxic, okay. Very simple study. So here you see, it is using the dihydrofolate reductase as we have already thought of. Then cellular tumor antigen P53, but along with that, see here it is also interfering with proton coupled folate transporter. Okay, I do not have any this thing in-depth info about this particular protein, but it seems like it is having a much more broad spectrum what you call impact. Okay, so there are some more also, as you can see. So now this could be good or bad depending on your analysis, but further research is also needed. So what does it mean? It means that the previous protein, a previous drug, is having a limited audience. On the contrary, this particular protein, this particular anti-cancer drug, methotrexate, might have a larger audience. Now this is the gross study. So further you have to study and figure out which one is less toxic, because you have to find out the each and every protein, whether it is a vital protein or it can also happen that it is also in a pathway of the metastasis pathway only. Okay, nobody knows until and because I'm not an expert in this particular drug, so I cannot comment. But what I'm trying to give you is an idea that how the preliminary ADMET analysis you can do. There are some specific steps in ADMET analysis, as many of you are already aware of, but for the beginners this particular tool is very, very important. Okay, so we discussed what? We discussed some basics of how cancer develop and how the intricacy of drug development are there. Okay, so subsequently we will study 2 more tools in this particular domain only, basically in this domain where structure based analysis will be dealt with. Okay, so